So next, we're going to talk about ASOs. We're going to move to the ASO section still in Pillar 2. So first, we have Ionis, who's going to be presenting an update on their program for ION 582 for individuals living with Angelman syndrome. We have Dr. Becky Crean, who leads clinical development for this AS program at Ionis. Becky.
Forward, backward. Thank you. Thanks, Allison. As Allison mentioned, I'm Becky Crean at Ionis Pharmaceuticals, and I lead the Angelman syndrome program back home. And thank you to FAST for inviting us to be here today. It's always a pleasure to come and spend the weekend with you and share updates and hear stories from you and talk about our progress in our program. So today I'll be able to do that with you and share some exciting news about not only the current program, but our upcoming plans as well. And I also have the pleasure of having Dr. Lynn Bird here with me. I think she's online somewhere, who's also going to give part of the update as well. And I think you all know Dr. Bird. She's a real leader and pioneer in the field. She's a principal investigator on our HALOS trial and is going to be sharing some data with you today. All right, so let's get started. These are my disclosures. Okay, so over the next probably 30 minutes, I'm going to be talking through a few different aspects of our program with you. So first, we're going to review, just briefly review the design and the primary study objectives of our current HALOS trial, which we've talked about in the past. It's currently ongoing. It's really our first in-human trial designed to look at safety and also some clinical changes in individuals living with Angelman syndrome. So that trial is ongoing. The first part of that trial, the first 6 months, called the MAD, is completed. We're going to share data with you today around that. And everybody who is eligible rolled into the long-term extension. So that study continues to advance. And then Dr. Bird is going to present those data results from the Part 1 MAD findings. She'll review some of the safety and the clinical data as well. And then I also have the pleasure of talking with you about our next steps in the program. And we're going to review the design of the pivotal phase 3 trial called the REVEAL trial with ION 582 and tell you a little bit about our plans and timing of that going forward. So a lot of things to talk about this morning. So I'll just take a step back and just review briefly about what ION 582 is. It is an ASO designed to stop the silencing mechanism of the paternal UBE3A gene. You've heard a lot about how that's done. I know it's not new to you, but just to review that it does knock down the antisense transcript in the paternal allele, which then upregulates UBE3A protein in the brain. This is a particular kind of ASO. They're not all the same. This is one that has certain types of chemistry and sequencing, and depending on those sequences and chemistries, they can confer different properties of the ASO. So there's a number of different types of ASOs out there. There's the 2' Mo, which is what ION 582 is. There's LNAs, and they all have different properties. So 2' Mos are ASOs designed specifically with the intention to achieve greater potency for knocking down the target of interest, reducing that target RNA, and they're also designed for their increased safety and tolerability. So it's for these reasons that Ionis chooses to develop 2' Mos as their ASO of choice when we're going into diseases into the brain. And we do have quite a bit of experience with this approach. We've been using this type of chemistry for quite some time, tested over 14,000 patients with this type of particular chemistry. We've looked at them in indications in spinal muscular atrophy, ALS disease, Huntington's, Alzheimer's, Parkinson, many others in development as well. So we have a very deep and good understanding of this type of chemistry and what it can and can't do in the clinic. So the HALOS trial, which we're going to talk about in terms of data in a moment, this is our first in human clinical trial. So it's the first one we go into after all the preclinical work has been done with this compound. And this is a trial that we started a couple of years ago. We've been able to provide some updates along the way, but just to review a little bit about the design and the goals and intentions of the study. And we've talked about this before. Each trial, each stage of development is designed to answer some very specific questions about the compound of interest. And so this particular one, the first in human, is really designed to assess the safety and tolerability of the compound. It's a global trial. It's open label, which means that everybody receives active drug. There is no comparator arm. We looked at 3 different dose levels of ION 582 in a really broad spectrum of individuals living with Angelman syndrome. So there were 51 participants in the first part of the trial, both male and female, and they ranged in age from down to 2 years old up to 50 years old. These individuals had either a deletion or a mutation genotype. So as we've heard before, ASOs are given intrathecally into the CSF with a lumbar puncture, and because of this type of procedure, the participants are sedated during the procedure. So the primary outcome, as I mentioned, was safety and tolerability, but of course we looked at a number of different outcome measures to look at change in important areas of clinical function. And we did it through a number of assessments, and that allowed us to really get a broad look at all the possibilities that ION 582 had to impact some of these important areas in clinical functioning, such as communication, cognition, motor function, sleep, seizures, activities of daily living, all of those things that are important to all of us. So the schematic in the bottom is what we're going to talk about today, which is just the part 1 portion of HALOS. HALOS is actually quite a long trial. It's got 3 parts to it. Part 1 is this MAD, where we looked at 3 different dose levels. We gave the compound at day 1, 1 month later, and then 3 months after that. So a total of 3 months with 3 injections. At the end of that 3 months, participants went into what we called a follow-up period, where additional drug wasn't given, but we continued to assess. And so the low dose, you can see, we had our last follow-up period was at 4 months, and we did assessments then. But in the medium and the high dose, we continued to follow them out for 6 months before giving the next dose. And so what we're going to present on today is that 6-month time point when everybody had that assessment in the medium and the high dose. After that, everybody moved into this long-term extension, and that includes parts 2 and 3, where essentially the medium and the high dose are continuing to be tested on all participants for up to 4 years, really. And that'll be a long-running trial as it goes on with the phase 3. So let me just talk with you a little bit about the ways we assessed clinical functioning in HALOS before Dr. Bird starts to talk about the data generated from these assessment tools. So again, I mentioned that we tried to touch on all the key areas of functioning in this trial, and we did that in sort of a 3-pronged approach. We had assessments that measured the participant directly, which is primarily done through the Bailey assessment, which I'm sure you're all familiar with. We also did it through parent perspective questionnaires, and we did that through the Vineland, and also the measure called ORCA that's specific for communication. And then we also asked the clinician, the doctor, what their perspective was on how clinical functioning may have changed over time, and we did that with something called the Symptoms of Angelman Syndrome Clinical Global Impression of Change. It's a mouthful, but it's basically asking the investigators their opinions on certain areas of functioning where they may have seen improvement or not. You can see on the left that there's a lot of overlap with these assessments in measuring the same functional areas. So like communication is measured with most of the tools, same with motor functioning. And we do that again so that we can get this broad perspective from the clinician, the participant itself, and then the parents on how they think things are going in the trial and any changes they might be seeing. So with that, I'll turn it over to Dr. Bird to talk with you about the results that we see at that 6-month assessment. And Dr. Bird, I'll just tell me when you want me to advance, and I'll do that.
Thank you. Hi, I'm Lynn Bird. I'm from the University of California, San Diego, and Rady Children's Hospital. And I've been involved in Angelman syndrome research since I was drafted in 2001 by Terry Jo Bichelle. And things have changed a lot since then, as you heard from our PODET's historical overview yesterday. Next slide. Here are my disclosures. I'm a consultant for Ionis in their Angelman program and a PI on all 3 of the ASO trials. Next slide. So as you heard from Becky, there were 51 participants enrolled in the Part 1 multiple ascending dose or MAD phase, and the ages ranged from 2 to 34 years and include mutation and deletion genotypes. Ages and genotypes aren't distributed evenly between the dose groups, which is expected given the way the study was designed and given that deletion is so much more prevalent than mutation genotype. So of those 51 participants, 50 completed Part 1, and all of the completers opted to roll into an ongoing long-term extension. Next slide. ION 582 demonstrated a favorable safety and tolerability profile in all 3 doses evaluated in the MAD portion of the HALO study. No discontinuation or adverse events were deemed related to study drug by the investigators, and most adverse events were consistent with the participants' medical history, symptoms of Angelman syndrome, or related to the lumbar puncture itself. The most common adverse events were fever, vomiting, and upper respiratory tract infections, in other words, the common cold. Additionally, there were no reports of lower limb weakness, ataxia, or radiculopathy. Next slide. Since HALOS does not include a placebo group, comparisons were made to natural history studies, abbreviated as NHS on the slides you'll see, which collected data over time in patients living with Angelman syndrome. So data from the medium and high dose groups were pooled at the final 6-month time point of Part 1, which increased our ability to detect treatment effects and homogenize the age and genotype data to enable matched comparison to the natural history data. Next slide. We'll first look at communication, since addressing these challenges is one of the most important unmet needs in Angelman syndrome. The 3 assessment tools shown here capture direct measurement by the Bailey on the left, subjective reporting by caregivers with the Vineland in the center, and clinician rating by the CGI on the right. In this and subsequent slides, the HALOS participants are shown in purple, and the black bars represent comparison data from the natural history study, so the change we would have expected over 6 months based on what we know from natural history. The graphs demonstrate consistent improvement from baseline to 6 months. For the Bailey and Vineland, higher scores means greater abilities, and for the CGI-C, a lower score means improvement. We don't yet have robust CGI data from the natural history study, so you don't see a black bar on the graph on the right. Next slide, please. These data show that receptive communication abilities also increase substantially at 6 months following treatment with ION 582. The Bailey data are shown on the left and the Vineland data on the right. Improvements in receptive communication in ION 582 far exceeded the minimal change expected in untreated patients living with Angelman syndrome. Next slide, please. Overall communication ability, assessed by caregivers using the ORCA, was also increased at 6 months following treatment with ION 582. The improvement shown meets the 2-point threshold considered clinically meaningful. Next slide. Substantial treatment effect of ION 582 was observed in the functional domain of cognition, in other words, learning. The graphs demonstrate improvement at 6 months in the Bailey compared to natural history data on the left, and the CGI demonstrates clinically meaningful improvement according to the CGI-C scale. The Vineland isn't shown here because it doesn't directly assess cognition. And not shown here is also that there was a statistically significant dose-dependent reduction in EEG delta power in children treated with ION 582 that correlated with improvement in cognition. Next slide, please. The CGI is made up of several different domains, and this slide portrays the percentage of HALOS participants that demonstrated improvement, which is a 1 point or greater change, and overall symptoms on the left, and in the individual domains to the right of that. You can see in every domain, the majority of participants showed improvement from baseline to 6 months. This was true for all domains assessed, including communication, cognition, motor function, as well as maladaptive behaviors, sleep, and activities of daily living. At the far right, seizures had the most modest response, but this was expected because the study protocol required that seizures be well controlled to start with. Next slide. Similar robust and consistent results were seen in a responder analysis of the Vineland, in which response was defined as improvement from baseline to 6 months that exceeded age and genotype matched natural history data. So importantly, I'd like to highlight the significant improvements in daily living skills. Next slide. Across the board, in totality, ION 582 demonstrated consistent response across all domains evaluated in the HALO study, with the majority of participants showing improvements in each individual domain. Next slide. And perhaps more importantly than what investigators thought, this slide showed what caregivers observed in the participants' ability to focus, to communicate, to move, sleep, and to take care of their daily needs. Green means better, yellow means no change, and red means a little bit worse. So for every area, at least 50% of participants were felt by their caregivers to show improvement. Next slide. In conclusion, data from part 1 of HALOS, the multiple ascending dose portion of the study, demonstrated a favorable safety and tolerability profile for ION 582 at all dose levels. EEG delta power was reduced after 6 months of treatment. There was evidence of clinical improvement across all key functional domains. 97% of participants showed clinically meaningful overall improvement in their symptoms of Angelman syndrome on the CGI. Improvements in communication, cognition, and motor function exceeded natural history data on the Bayley-4, the Vineland-3, and the ORCA. And benefits were seen in all ages and both genotypes. These data, while early, are encouraging and warrant further clinical development and evaluation of ION 582 in a well-controlled phase 3 trial. And with that, I'll turn it back over to Becky to talk about next steps for ION 582.
Thanks, Dr. Bird. And don't go away because I think you're going to be on the panel later. So as Dr. Bird mentioned, we are very encouraged by these results, and they really helped Ionis come to the decision that we needed to continue development with this compound. And so with that, we designed a phase 3 trial called the REVEAL trial that I'm going to share some details with you about that now. And while the design was really informed by the data generated from the HALOS trial, it was also heavily informed by talking with you, the community, the parents, the clinicians, and really trying to put together a program that would give us the greatest chance at success in showing what ION 582 can do in individuals living with Angelman syndrome. So it really was a collaborative effort to put this program together in a way that we think will be able to show what this compound can do. So I'll share some of those details with you now. So this is a trial called REVEAL. It's a global phase 3 trial for children and adults living with Angelman syndrome. This is a pivotal registration trial, and what that means is that the data generated from this trial will be used by regulators to make decisions on whether this compound should be commercially available or not. So it's a really important trial. We have to really kind of tighten up all that we do in it, pick the right assessments, pick the right dosing regimen so that we can give this the best chance it has to be able to convince regulators that it should be available for use. So the HALOS trial that we just talked about, as I mentioned, the primary objective there was really understand safety and tolerability because it was our first trial in patients. This trial is a phase 3, so while safety is still a primary objective, and we'll continue to measure that, we're also going to be measuring a number of clinical outcomes assessments to determine efficacy. Efficacy meaning, does the drug make you better or not? So to do that, we'll look at measures that assess communication, overall disease severity, cognition, sleep problems, motor function, activities of daily living, and we're doing that with some of the similar assessments that we used in HALOS as well, some that are very familiar to you, the Bailey, the Vineland, the ORCA, the CGI. So this trial, for participants in it, will last about 3.5 years if you're in the trial. And so what that means is it's broken up into 2 parts. The first part is it will be the double-blind, placebo-controlled portion of the trial. That'll last for about a year, and that's when individuals will receive either ION 582 or a placebo comparator. So once you get through that first year of the double-blind, placebo-controlled portion, then everybody will receive open-label drug in a long-term extension. That'll run roughly 2.5 years for each individual participant, and that's where we'll get more long-term safety and efficacy data with the compound. So again, about 3.5 years total for an individual family. The first year would be the double-blind portion where nobody knows which treatment they're on, and then everyone will get open-label drug in the long-term extension. So I mentioned 2 things, double-blind and double-blind placebo-controlled. And so what does that mean exactly? So double-blind, we heard from Jennifer earlier this morning, just means that nobody's going to know which treatment arm they're in, right? So the clinicians will be blinded, the families will be blinded, the sponsor will be blinded, all the study staff will be blinded. And that really is so that we can really determine what effects we're seeing aren't based on our opinions, but they're based on the data that gets generated in the trial, and everybody's blinded to that. And then the second thing I mentioned is placebo-controlled. And again, Jennifer talked about that this morning and gave a really nice description of the different types of control and comparator arms there are. In this trial, this will be a placebo-controlled trial. So what that means is that participants in the placebo arm will have exactly the same procedures that you would have if you were in the active drug arm, meaning you still get a CSF draw, you get a lumbar puncture, you get sedation, you get an injection, but it's not with the active drug, it's with a placebo. Everything is the same. And the reason we chose this is we feel that this is really first of all, it's the gold standard in determining safety and efficacy in a compound. But we also feel that this is the most robust type of design that's scientifically rigorous, that will really answer that question for us of whether the compound is better than no drug or not. So what does the trial look like? Again, I mentioned it's a global trial. We'll have sites across the globe. It'll be evaluating the safety and efficacy of 3 dose cohorts in this trial. What that means is 2 dose levels of ION 582 will be studied, and then the 3rd cohort is placebo. And again, everybody's blinded, they won't know which one they're in. We'll enroll about 200 male and female participants. Again, a very broad age range from 2 to 50 years of age. Both deletion and mutation genotypes will be enrolled in the trial. So all participants will receive the intrathecal injection with a lumbar puncture. It'll occur every 3 months, and participants will be sedated while they're undergoing the procedures. Again, I mentioned that we'll continue to assess safety, but we also look at efficacy in this trial. And what we have heard from the community is that communication is the #1 area of concern and the most important for a treatment to address in Angelman syndrome. So we have chosen as our primary endpoint to measure expressive communication, and we're going to do that with the Bailey 4 scales of infant and toddler development. And again, you're all very familiar with that. We've heard talks about that over the week. But in addition, we'll continue to explore all the other key areas of function that are important. Changes in overall disease severity, cognition, other types of communication, sleep, motor functioning. We'll assess it all in this trial as well, but the primary focus will be on communication. So you can see the schematic at the bottom that will test 2 dose levels and placebo. So part of the participants will be in the 40 milligram group, part will be in the 80 milligram group, and then part will be in the placebo group. With drug delivery every 3 months. At the end of that 12 months, that first year, we'll look at that primary outcome assessment and expressive communication. But while we do that, all the participants will roll into the open label long-term extension. Again, that'll run for about 2.5 years for each participant, where they will receive open label either 40 or 80 milligrams every 3 months. So just to provide more detail about what that would look like for an individual family who might participate, there'll be a short screening period where you fill out some questionnaires to determine if you're eligible to enroll in the trial. If so, then you move into the blinded placebo-controlled period where you'll be assigned to either the 2 dose levels or placebo. Whichever one you're on, you'll go through exactly the same procedure. So you'll get drug delivery every 3 months, where you'll come into the clinic for that. You'll do that for about a year, but in between those visits, we'll also have you fill out some assessments at home as well. We'll continue to look at important areas of functioning both when you're in the clinic and then when you're at home as well. And again, once you finish the double-blind period, then everybody rolls into the open label extension at the end of that. And so I mentioned that this was really designed, data-driven from the HALOS trial, but a big part of it was hearing from the community on how best to design this trial and to focus on things that are most important to you, design a trial that's reasonable to participate in. We know these are burdensome and you have to take time out of your days to do this. So we took a lot of feedback from the community, and we really feel confident this is a very scientifically rigorous and robust trial that will really help us answer this next question. So where do we go from here? We've aligned with the FDA on our trial design. We will get final global regulatory input by the end of the year. And we will continue to advance the HALOS trial, as I mentioned. All the participants that are in there will continue receiving ION 582. And this is really helping generate long-term safety and efficacy data that'll help support ION 582 in the future. We do plan to add some cohorts to that trial. We'll start off with adding an infant cohort, a small infant cohort, age 0 to 2-year-olds, to start to get an understanding of the safety and tolerability of the compound in that really young population. We are on track to start this pivotal phase 3 trial called REVEAL by the first half of next year. So we have a lot of work to do back home at Ionis, and we hope to continue to give you updates along the way. But with that, I would just like to express my sincere appreciation for, first and foremost, the families in the community in the HALOS trial. Your participation got us to where we are today, but also to the entire community, the sites that participated in HALOS, the study staff. Collectively together, this collaboration has helped us answer some really important first questions and helped us move this program to the next stage, which is the pivotal registration trial. So with that, I'll say thank you.
Time for questions, yeah. Sure. We have about 5 minutes for questions, okay, if we want to get some of them here before the panel. Do we have any questions in the audience? I have 1,000 questions from online, which I'm happy to start on, but I'll give you all a chance first. Dr. Bodette? Someone
Hi.
Oh, go ahead. I'll give this to Art after you. You go ahead.
So I have a question as far as the outcome assessment being only Bailey for expressive communication. Mm-hmm. So with that being said, I know all of our children are enrolled in speech therapies, but is there a specific amount or type of speech therapy that would be required to ensure that we are working with our children that are in the participants to ensure that we look for significant change if that is our only outcome measure? Will there be a required amount of physical therapy per week? Or I'm not physical therapy, speech therapy for the participant?
No, we aren't requiring any particular type of outside therapy to be in the trial. Obviously, they're on a number, your children are on a number of different types of therapy modalities, and whatever they're on is okay to come into the trial with. To be in the trial, you wouldn't have to change anything that you're already doing. Having said that, we would ask that whatever you are doing, you hold consistent and constant throughout your time on the trial, so that doesn't become a thing that changes, that may impact what we see in the data. So we just ask that you hold that constant and keep doing the same thing you've been doing, just continue that through the trial.
Question here. On the UPD, over here, on your right.
Thanks.
Yeah, thanks for that talk, it was really brilliant. Just on the UPD front, would that be then looked at as part of the HALOS trial? I saw that you were extending to younger kids through that. Is that the mechanism that you're going to try to use to go out to further genotypes? And if so, when are you going to do that?
It probably would be the same mechanism. We've built that foundation in that trial, and it's a good source for us to continue to evaluate different aspects of the compound in different populations. And so yes, we do know we're going to start fairly soon with the 0 to 2-year-olds, but that would also be the right place for us to expand into different genotypes. So we don't have the specifics of that worked out yet, so I couldn't share more than that, but just to know that we are working through those discussions and coming to where we can bring those into the trial, and it likely would be the HALOS trial.
Great, thanks. And if I could just ask one follow-up question. For delivery or manufacture and supply of the drug in Europe, have you got locations for that, and what countries are you using? I'm from Ireland, so I just want to know if I have any leverage with our health authority.
For manufacturing or for?
Yeah, manufacturing and distribution.
So right now we manufacture in Carlsbad, California. We do all of our manufacturing there and distribute from there. So as far as I know, that's where it will stay.
Okay, great, thank you.
This is very, very encouraging. I have 3 very quick questions. First, you talk about sedation. What percentage of the patients are able to get away with less than general anesthesia?
So we allow any type of sedation, and it's left to the judgment of the principal investigator on what level they think is warranted to help the kids during the procedure. We have heard from Dr. Barry Kravitz that she's gotten to the point where she's not having to use sedation on everyone, and then all the way through to general anesthesia, and it's everything in between. It really is individually based, and we allow that flexibility in the trial.
Is it fair to assume that a large fraction get general anesthesia at the moment?
I don't think so. Not in our data. We're not seeing that split.
My second question is, what's going to be the ratio of patients in the placebo control? What's the ratio of low dose, high dose, and placebo?
It's a 2-to-1 randomization whether you get active drug or placebo. But drilling down further than that, because we are studying 2 dose levels and placebo, it would be what we call a 1-to-1-to-1 randomization. So a third will get the 40 mgs, a third will get the 80 mgs, and a third will get the placebo. But overall, it's a 2-to-1 active to placebo randomization.
And one final question. What's the current nominal cost of SMA treatment, and is it reasonable to assume that Angelman treatment could be in the same order of magnitude?
Oh gosh, I'm not on the finance side. I honestly can't answer questions about cost. I'm not sure.
I have a few questions from online. Many are coming in, but I'll ask just the first 2. The first question is, why were the loading doses removed?
From you mean from what we studied in HALOS versus what we plan on doing in the REVEAL trial?
From the original.
Yeah, so in the HALOS trial, We primarily did that single loading dose. You saw we did baseline and then 1 month and then 3 months. We primarily did that single loading dose really to get us a better understanding of safety and tolerability in a very short period of time. It was also to help us characterize the PK properties as well. I mean, we were going into this without any data in humans, and so it was an opportunity to collect more data. But from that data from the HALOS, what we've learned is that we don't need that loading dose going forward, so we dropped it.
The second question is, what placebo is injected for the placebo group?
That's a great question. So I'll talk about the active compound first and how we administer Ion 582. And within the solution, there's a small amount of drug, and then the rest is filled up with artificial CSF. When you withdraw CSF, you want to try to replace it in the system. And so we have a studied drug that includes both active drug and artificial CSF. And that'll stay the same in the REVEAL trial as well. The placebo will be made up of just the artificial CSF. So it's exactly what's in the drug compound, just without the active drug.
Hi, I'm here. I'm Stephanie. I'm mother to Asher, who's 2 years old, and we live in Hong Kong. As you mentioned, it's a global trial, so I was wondering if there might be a chance for the trial to expand into Asia.
There is. We're actually doing feasibility across the globe, and it includes Asia and countries in your area. So we're collecting that feasibility now and making site selection. So we do have plans to be in that area.
Thank you.
Mm-hmm.
We're going to have a whole panel where there's going to be lots of time for questions. So I think we're good to get started on the next talk, but I promise you all, you will have plenty of time for questions. Thank you, Beth.
Do you want this?